So this lecture video is on cell division, particularly the cell cycle and mitosis. So what's the point of cell division and why cells reproduce? Well, we need cells to be able to divide so that we can have cell replacement and also growth as well via cell division. So here is our early human embryo, and we start off as an egg and a sperm, and they fertilize. The sperm fertilizes the egg, and it's called a zygote. And then this fertilized egg divides and divides and divides through cellular division. And as it divides, it specializes, and this is called cell differentiation. And so certain cells are going to do certain functions, but without this cell division, we wouldn't be able to go from one single fertilized egg into a human being. It's a pretty cool process. Through the cell division, this is how we're going to get different types of organs. It's just remarkable how one cell can divide and create a human being. It's pretty cool. So cell division is really important for growth and development. And also cell division is important for replacing old cells, cells that may have been lost due to damage, or maybe they're short-living cells and so they need to be replaced. So that's important as well. In a previous lecture, I talked about how cells come from other cells. This is one thing that's free in life. You don't have to run down to Walmart and get a package of cells, right? When you scrape your knee or your arm, you get a cut on your arm. In a few short days, your skin is back to normal. You have a little scab that might form, it falls off, and you're back to normal. And that's all thanks to cell division. And I talked about reproduction as well. All right, so there are two main phases of the cell cycle. The two main phases are interphase and the M phase. Interphase consists of smaller phases. This would be G0, G1, S, and G2. A lot of people want to put G1 and G2 beside of each other, but don't forget there's an S phase in between those two. So G0, this is when the cell is at rest. G1, the cell grows and develops. The S phase is probably one of the most important phases of interphase, because within the S phase, we have DNA synthesis. And remember that synthesis is just a fancy term for make. So here, we're going to copy the DNA. Make copies of that DNA. And then in G2, we're going to prepare the cell for division. And then we move into the M phase, which is mitosis. And this is where the process of cell division, or where the cell divides. And we're going to talk about prophase, metaphase, anaphase, and telophase, which are all phases of mitosis. Please know, interphase is not a phase of mitosis. It is the first phase in the cell cycle. It includes G0, G1, the S phase, and G2. All of those are smaller phases that make up interphase. Okay? For mitosis, remember PMAT. PMAT. And cytokinesis is the actual separation of the cells, which we call daughter cells. I think I put an extra. Sorry, guys. So let's draw out the cell cycle. Here is G0, where the cell's at rest. Then we have G1. This is where the cell is going to grow and develop. Then we have the S phase. And in the S phase, we're going to make copies of DNA. And then we're going to have G2. This is where we prepare it for division. And lastly, we're going to have the M phase or mitosis, where the cell is going to divide. Now then, I want to show you what it's going to look like. So let's look here. This is our parent cell. Okay, so this parent cell is going to enter at G0. It's going to go through G1, where it's going to grow and develop. And then in the S phase, we're going to make copies of the DNA. So when it leaves the S phase, it's going to look something like that, right? We've made copies. We're going to prepare it for division. And what's going to happen is out are going to come our daughter cells from mitosis, and you're going to see you have two new daughter cells that are identical to the parent. And that's because we made copies over here in the S phase. So let me show you what it looks like in blue. If we have the same parent cell go in, but we don't copy correctly in the S phase, it doesn't copy. If we didn't make copies, what would happen is we would end up with a cell, with two cells that didn't have enough DNA. It's kind of like if I wanted to get notes from you for a test, and maybe I missed class that day, so I need to get notes from you. I'm not going to take your sheet of notes and rip it in half and give you one half and give me the other half. That's not going to do either one of us good, right? You're not going to be able to be prepared for the test. You're missing half your notes. I'm not going to be prepared for the test. I'm missing half my notes. well, the notes you took for me. Anyways, so what we need is another full copy of the notes, right? So you would either want to like scan it, go find a copier, and then give me those notes. So that's why the S phase here is so important. It's because otherwise, if you don't copy the DNA correctly, and here in my drawing, the DNA is the smiley faces. But if you don't copy it correctly, what happens is you end up with these blue cells down here that don't have the correct amount of DNA. They're not going to be able to continue. They're not going to be able to divide. They're not going to be viable. They're just not going to be able to function because they don't have the correct DNA. So the S phase is really. All right, so here's just another drawing. I know that my artistic ability is really lacking, so here's another diagram to show you what the cell cycle looks like. And also, the thing I like about this one is it also shows you interphase here and the M phase as well. So it shows you how G0, G1, S, and G2 are all part of interphase. All right, so let's talk about the phases of mitosis. So we have PMAT. There are 4 phases of mitosis. I want to start by showing you the parent cell. So I'm going to attempt to draw chromosomes, which is where DNA is going to be found. It's like a tightly wound structure. Here's my best attempt at chromosomes. So I have a blue chromosome and I have a purple chromosome. Sorry guys, that's my dog. Okay, so after this parent cell has gone through interphase, which remember is G0, G1, S, where we've copied the DNA, we need to show that we've copied the DNA. So this is one that's been through interphase. So here's the parent cell. It went through interphase, and now it's going into the first phase of mitosis, which is prophase. So I'm going to draw 2 blue because we made copies of the chromosomes, right? And 2 purple. All right, so now we're going into prophase. So in prophase, what's going to happen is the chromosomes are going to condense. And what are called spindle fibers are going to form. So here, chromosomes condense and spindle fibers form. This is also known as the mitotic spindle. So then we're going to move into the next phase, which is metaphase. And in metaphase, the chromosomes align over the middle. So here are the mitotic spindles. And they're going to move the chromosomes to the middle of the cell. So think meta-middle. All right, that's as good as it's going to get. Then we go from metaphase into anaphase. And this is where the sister chromatids are pulled apart. So I think anaphase apart. And what are they pulled apart by? They're pulled apart by that mitotic spindle or those spindle fibers. So here, and they're pulled apart, and then I want to put two opposite poles or sides of the cell. So here they're being pulled apart. And I sometimes think of this as A, where the chromosomes are, N for the middle, right? And then A, you'll have chromosomes on either side of nothing, right? So the N stands for nothing in the middle. You have chromosomes on either side. Anaphase, and then the last phase, if I have room, is telophase. Whoops. And in telophase, two new daughter cells are formed. And they're genetically identical. And so this here is called cleavage. Basically, the cells are going to pinch apart. And that final division is called cytokinesis. So here I'll draw the two new daughter cells. Hopefully you can see that blue. And you can see how they are identical to the parent cell. It doesn't matter that the purple is on the right and the blue is on the left, but you can see they have one of each right in their cells. And there are 2 of them. That's important. At the end of mitosis, there are 2. I think that just actually made it more confusing that I drew arrows. So let me erase those. But you can see there are 2 cells there. And because this is so everywhere, here I'll put start of mitosis. And this is where we end, down here, with the 2 new daughter cells. So it's prophase, metaphase, anaphase, telophase. Hopefully you can read all the scribble. If not, don't worry, I have excellent diagrams for you. So here's interphase. All those squiggly lines are the chromatin that's duplicated. And then here in prophase, you have the chromosomes condensing to the middle. You also see those mitotic spindles form. In metaphase, you can see how they align over the middle of the cell. And in anaphase, they're separated. They're pulled apart by those mitotic spindles to either pole or opposite sides of the cell. And then there's telophase, where we're forming 2 new cells with the nuclei in the middle with the same genetic information as the parent cell. And you can actually see like cleavage furlough or where those cells are pinching apart. And that's how we make new cells. Here, cytokinesis, or that cleavage furlough, and you can see how they pinch apart into 2 new daughter cells. And then lastly, I want to talk about cell division. It's pretty tightly controlled. So there are checkpoints, remember, the cell cycle where we have G1, S, and G2. Well, there are checkpoints between G1 and the S phase, and there's also a checkpoint between S phase and G2. And what those checkpoints there are for is to make sure the cell grew and developed properly, the DNA was copied properly, we don't have any funky mutations in the DNA, and then we prepare it actually for division. It's ready and to be prepared for division. So basically they just check and make sure that everything in the previous phase has happened correctly. But sometimes things can go wrong with the control. And that's when cell division occurs sometimes, even though there are abnormal cells. Typically, the body can pick up, the cell cycle can pick up on abnormal cells, and it will try to get rid of those cells. It might actually tell the cell to kill itself, or to terminate itself. That's probably a better language there. Sorry, guys. It'll tell it to terminate itself because it is abnormal, and we don't want abnormal cell division, because abnormal cells that divide is known as cancer. So how do we end up with these abnormal cells? Well, what happens is the DNA becomes damaged. And it can be due to a few things. Chemicals, heat, and radiation can all affect the DNA within the cell. And so you're going to have issues when that cell is copied. And having this damaged DNA is going to cause abnormal cells. So let's talk about the difference in benign tumors and malignant tumors. So a tumor is a mass of these abnormal cells. Benign tumors are located in one area of the body, or localized. So they are localized to one area of the body. Malignant tumors, they are actually going to travel. So the cells, I'm going to put here, I'm going to erase this real fast. Malignant tumors, the cells travel to multiple areas of the body, and then they do that via the bloodstream. And then they actually hop out of the bloodstream and start to divide in the other part of the body. So malignant tumors also secrete chemicals that cause blood vessels to actually grow to them. And when blood vessels grow to these malignant tumors, that's going to provide these cells, these abnormal cells or cancer cells, with more nutrients and oxygen to do even more cell division and to divide it much more quickly. And so malignant tumors, because of that, because they secrete these chemicals that increase blood vessel growth, they actually are going to grow rapidly because they're getting more nutrients and oxygen. And a lot of times they outcompete your normal cells for these nutrients. And that can be how organs begin to shut down, unfortunately. My mother had breast cancer. She had stage 1, and they caught it pretty early, obviously. And the first thing they did was check her lymph nodes. So they're found like under the armpit, and they checked her lymph nodes to see if there were cells there, cancer cells, because if there were, they knew then that the cancer cells in her breast had actually traveled to a different part of the body. And this is unfortunately how someone that might have breast cancer ends up with ovarian cancer, uterine cancer, or a different form of cancer, because these cells tend to travel throughout the body. And like I said, they're very aggressive. They grow very rapidly, and it's very unfortunate. So it's really important to be scanned for cancer if you know that, or to get those annual scans. Like my mom needed a mammogram, and she actually almost thought about not going, but it's really important to do so because they can grow so fast and outcompete your normal cells for those nutrients and oxygen, and kind of end up outcompeting and damaging those cells. So the other thing I wanted to mention was the chemicals, radiation, and heat, and how they can damage the DNA. There was a guy, if you read about it, there was a guy who used Roundup which is a weed killer. He was part of a maintenance crew at a school, and he actually used Roundup to every day, pretty much. I'm not exactly sure how often, but he used it quite often to actually keep the maintenance of the grounds at the school. And so exposure to that chemical, over time, he actually ended up with cancer. And it was found that the cancer he had did result from the chemical in Roundup, and he was awarded millions of dollars because of that. So exposure to chemicals over long periods of time, certain carcinogens can damage your DNA and result in cancer. But there are treatments. So we have radiation and chemo, and the biggest difference between the two is how aggressive is your cancer? What stage are you at? And has it really moved, or has it become malignant? Do you have a malignant tumor? So with radiation, this treatment damages the cancer cells in one part of the body, or specific So this is what my mom had. Sometimes you can have both. So a patient may be prescribed both radiation and chemo. My mom, luckily, because she had stage 1, she just needed they were able to remove it surgically, and then she had radiation to make sure that they suppressed growth in that area and damaged those cancer cells. And then chemotherapy, this is going to be used more often in aggressive malignant tumors. It's going to target the whole body, and it actually kills or slows the growth and division of healthy cells. And so that's why a person that has radiation will typically not see hair loss. But someone that has chemotherapy will see hair loss because it does affect those hair follicles, which are growing and dividing. And so it does slow the division of both the healthy cells and cancer cells, because it's trying to target more than one part of the body. And like I said, that's typically with a more aggressive or malignant tumor. And lastly, here is a diagram for you. So here's how we start off with one of those abnormal cells. Our DNA in this cell has been damaged due to radiation, chemicals, or heat, maybe a combination of those. And then here is the tumor. At this point, it's still a benign tumor. You don't see any blood vessel growth. It's just growing. No movement of the cells yet. Here, you can see this malignant tumor has blood vessels that are growing into it, because it's secreting chemicals that are causing the blood vessel growth into the tumor. And you can see how the cells are actually hopping out of the tumor, moving, dividing out of the tumor and into the blood, and then the abnormal cell here is escaping from the blood vessel where it's going to start a secondary tumor downstream. The last thing I'll mention is if you watch Grey's Anatomy, there is an episode where they choose to 3D print someone's tumor that they're trying to remove because it's so large and it's entangled in so many blood vessels that they were trying to 3D print it so they could get a better idea of how to remove the tumor successfully because of the amount of blood vessel growth that had occurred within this tumor. And so what was going to happen is if they tried to just take out the entire tumor, the person would actually bleed to death because you would sever so many of those blood vessels. So they're trying to figure out a mechanism of how to remove the tumor around those blood vessels. And on that sad note, I'm going to end this lecture video. Please let me know if you have any questions. Thanks, guys.